Hi, I'm Alexis Reece, and my research is on the metamorphic success of sea star larval clones. When you think of a sea star, you probably imagine the familiar five-armed critter pictured at the bottom left here. However, this is only the adult phase of the animal. Sea stars begin life as eggs and sperm drifting in the water. Fertilized eggs develop into planktonic larvae, which then metamorphose back to the seafloor as miniature juvenile versions of the adults. Prior to metamorphosis, some larvae undergo cloning, a form of asexual reproduction in which a larva splits apart to form an anterior head clone and a posterior body clone. Each half is capable of regrowing its respective missing piece to form two genetically identical individuals, which theoretically can metamorphose into juveniles. Cloning allows sea star larvae to multiply rapidly when conditions are favorable, thus maximizing the number of offspring produced and that may survive. As sea stars play key ecological roles in their ecosystems, a small increase in adult population size due to cloning could have an enormous impact on large-scale patterns in marine ecology. While the frequencies of cloning are known for some sea star species, information on the rates of metamorphosis and survival of clones is lacking in the literature. Only one paper has recorded clone metamorphosis, and that was of a single clone. However, without knowing how frequently clones reach the juvenile phase, we cannot say whether or not cloning impacts adult population size. That is where my research comes in. My central question was this: do clones reach metamorphosis frequently enough to contribute to adult population growth? To answer this question, I cultured larvae of the sea star genus Asterias, measured the frequency at which the larvae cloned, and tracked the development of head and body clone pairs to metamorphosis or death. We found that on average 5% of Asterias larvae cloned. However, only 23% of head clones and 73% of body clones regrew to the point where they were capable of metamorphosing. Since the summed success of both head and body clones is less than 100%, there was no net increase in the number of juveniles produced, suggesting that cloning does not occur to increase population size. Additionally, we found that clones that began to regenerate at all were almost certain to metamorphose into juveniles. If they begin to regenerate at all, they are likely to be metamorphically successful. This work leads to a bunch of other questions. First of all, what advantage does cloning provide, if not to increase sea star population sizes? What determines whether or not a clone will regenerate? What are the gene pathways involved in the regeneration process? Further research is needed to investigate these questions. Thanks for listening.